And from the Division of Cardiology, Dr. Kerry Stewart. He's a professor of medicine here at Johns Hopkins in the cardiology division, and he's the director of the Johns Hopkins Clinical and Research Exercise Physiology. And Dr. Stewart's with Dr. McFarland have been really developed the vision of this center, and he is on our planning committee and has been research. You're going to hear from Dr. Bamman afterwards from University of Alabama about the sort of national level exercise projects, and Dr. Stewart's the Johns Hopkins representative to that group. So without further ado, Dr. Kerry Stewart, thank you. So thank you. Thanks.
We did test it. Thank you. It's not easy for a Mac user to use Windows. Before I start with my talk, let me also say that, there's a big focus on precision medicine, and a lot of it will involve genetics. But our vision is we will rely a lot on gene therapy rather than gene therapy. So different approach, and hopefully a very beneficial approach. So my topic today is on exercise as it affects cognition. Now, we were funded by a Johns Hopkins University Discovery Award, and the formal title was Human Performance Role of Active Exercise in Enhancing Cognitive Function. And the design of this award was to bring together people from different disciplines. So the team consisted of myself, Peter Searson from Material Science and Engineering, Howard Egath from the Department of Psychology, and Marlene Williams, who's in cardiology. And the reason why I'm mentioning the multidisciplinary aspect of it is that we hope within this center, similar projects could evolve. Groups get together and apply for grants, things like that. So we think that's hopeful. So let me get on to the topic of this project we did. It just finished recently, so it's still a work in progress in terms of the analysis. But one of the purposes of today's program was not necessarily to present completed research that's ready for publication, but really to showcase the kinds of activities that are ongoing or planned or clinical programs that are exercise related and related specifically to the notion of prevention and management of a chronic disease. So exercise may, in fact, be a very natural therapeutic intervention for cognition by several mechanisms: delaying age-related cognitive decline, enhancing brain plasticity and learning, improving various aspects of cognitive function, regulating various blood biomarkers like DF and insulin-like growth factors, which are associated with brain function. We're also interested to see if there's a reduction in inflammation, which does happen with exercise. That may affect cognition, and also increasing blood flow. Data on the role of acute exercise, a single acute bout of exercise on cognitive function, and that's our interest for this particular project, are lacking, but could impact health and well-being, learning, and performance. For example, in mission-critical jobs like the military, the police, and outer space, where individuals need to not only perform at a high level of physical effort, but also need sharp cognitive function. So the overall aims of this study was to examine the change in cognitive function after a bout of exercise, to see which aspects of cognitive function are affected, if change in cognitive function is associated with exercise intensity, and also influenced by baseline measures of fitness and body composition, and also to look at the changes in blood markers related to brain function. I'm going to go into this a little bit more detail later, but the overall design was we started with a screening by telephone. Visit 1 consisted of informed consent, blood pressure screening, a measure of habitual physical activity, anthropometrics, DEXA, muscle strength, and oxygen.
Have your attention, please. All clear, Bloomberg 12-4. I say again, all clear, Bloomberg 12-4. All clear, Bloomberg 12-4. Thank you.
Thank you. Which characterized fitness and body composition, and we let the subjects practice the cognitive tests, which were all administered on a computer. The second visit consisted of another blood pressure screening, and then you see I have marked A, B, C, D, and then at visit 3, E. At visit A, which was the baseline, we took the blood, did cognitive testing. Then the intervention took place, which I'll explain later. Jesus Christ. And then 1 hour, 0 hour after exercise, 1 hour after exercise, and 2 hours after exercise, we drew the blood again, did the same cognitive testing. And then subjects were required to come back a day later, again, for the same battery of tests. The subjects were 60, males and females, 18 to 40 years old, BMI between 18.5 and 35, which is normal to mildly obese, no hypertension, not smoking, no diabetes, no contraindications to perform moderate or hard intensity exercises assessed by the PAR-Q Plus survey, which many of you are familiar with. They also needed normal or corrected normal vision, otherwise they wouldn't be able to see what was going on the screen. Females could not be pregnant, which we did urine testing because they undergo a DEXA test, which involves an X-ray. And no substance or alcohol abuse by self-report. Some of the measures for fitness and body composition were height, weight, circumference. We did the rapid assessment of physical activity, which characterizes habitual activity on a 1 to 7 scale. We drew blood for BDNP, IGF-1, cortisol, and a whole variety of inflammatory markers. We used DEXA for bone, fat, and fat-free mass. We tested muscle strength using a Biodex dynamometer for knee extension and flexion. And we did a VO2 max test where the subjects walked on the treadmill at 3 miles an hour. We increased the grade 2.5% every 3 minutes, and we measured their oxygen consumption till they could no longer go on. So now I'm going to go through some of the cognitive tests that we used. So one test was the Stroop. Let me just say that when I took these tests myself, it was not easy. But I was assured by a psychologist that many of these tests are relatively not easy, but appropriate for a younger group. And many of them are also used, though, in people who are older to see the extent to which they have cognitive impairment. So the Stroop was a test of executive function that measures selective attention, cognitive flexibility, and pre-potent response. So the way the test works is subjects are asked to identify the color. They have to name the color of the word that we show them. We only showed one word at a time, but I put it this way to give you examples. So for example, the word red may come up, and we ask them, what is the color? So this is pretty easy. But then on the next line, we might say to them, what is the color? Oh, I'm sorry. Yeah, what is the color? And so they have to get the right color despite the fact that the word is blue. So we did that. These are all called either congruent or incongruent responses. And then we use a lot of neutral words like burger, budge. What's a budgie? anyone know what a budgie is? I know what a wedgie is. I don't know what a budgie is. You get the point. So the idea was to see if they can concentrate enough to name the color, despite what the word is. Another test was the flanker test, or I call the arrow test. This is a test of executive function that assesses the ability to strategically prepare for forthcoming distractors when one is not even present. So the subjects are first asked to fixate on a plus sign in the center of the screen. Then an arrow will pop up pointing either up or down, and they just have to say, is it up or down? Under another condition, we will then show something like this and ask them, what direction is the center arrow? But in the incongruent condition, we then show this and ask, what is the direction of the arrow? And the purpose is to see if these distractors interfere with the answer that they give. We also did a verbal fluency test, which tests the ability to retrieve well-known information out of long-term memory. So the subjects are asked to recite out loud as many words as they can think of that begin with a certain letter. For example, G. And they had 1 minute to name as many words as they could think of with the letter G. And we were tape recording this and analyzed it after the fact. And we used 5 different letters that were randomly assigned during the 5 different test periods. The visual working memory test, which is short-term memory, is challenges the ability to detect change as a function of visual short-term memory. So the subjects are first shown a square with different colors in the little squares. This is only shown for 100 milliseconds, so it really just flashes on the screen. You see this. We wait for 900 milliseconds, a little less than a second, and then we show them this and ask them to identify which color has changed. This test was the irrelevant capture test, and it measures the ability to filter out irrelevant distractors. So first the subjects are asked to fixate on a plus sign. Why is this? On a plus sign in the center of the screen, as indicated here. Then 4 alphanumeric characters appear in a square matrix around the plus. So we would show an A, a 2, a 5, a D, and they had to identify whether it's a character or a letter or a number. But within 25% of those overall trials of 150 trials, we'd flash for a short period of time an image of something else, like a clock and high-calorie food or low-calorie food. And the reason why we use these foods is because those high-calorie foods, in fact, have been shown to be a much stronger distract than low-calorie foods. So that's why we call this also the food capture test. Now, there was also a long-term memory encoding task. In this task, it's a test of the repetition detection to make sure that subjects can pay attention to each object long enough for it to be encoded in memory. So at baseline, we showed them 180 images of common objects shown on the computer screen for 3 seconds each. So it could have been a car, it could have been a banana, various things like that. In addition, 15 individual images are shown twice with varying delays between the exposure. At visit 3, which was now 24 hours later, the subjects were again shown all the images from baseline, but they had to detect whether or not it was an item that was repeated. So it's trying to see if they could remember things after being exposed 24 hours later. Here's the intervention. So we had 60 subjects. They were randomized to one of three conditions. Rest, sitting in a chair, reading quietly, or using a mobile device, but no phone calls. Or mild exercise on a treadmill, where we adjusted the treadmill speed and elevation to a target heart rate corresponding to 50 to 70% of VO2 max, which we had measured on visit 1. Or hard exercise, where the goal was to keep them in a range of 70 to 85% of their VO2 max based on heart. For all conditions, heart rates were continuously monitored using a single lead ECG, and the exercise heart rates were monitored continuously. And as I mentioned, the speed and elevation were adjusted to try to maintain their heart rate plus or minus 10 of that target that we picked. So if someone had a target of 140, if their heart rate went below 130, we'd make the treadmill go faster. If it went above 150, we'd try to slow it down, and so on and so forth. Here's an overall view of the subject characteristics, and don't pay attention to all the details, but I'm just going to say that in all three groups, the hard, the mild, and the rest group, we did the calculations of differences between men and women. In most cases, they were different because there are gender differences, but we knew that. Overall, the age was about 25. The BMI was 24. Weight was about 153. And the VO2 max was 44, which is about average for that age group. We also did knee extension and flexion. So in terms of did the intervention work? Well, at rest, we didn't measure the heart rate, so whatever happened, happened. In the hard group, 100% of the subjects stayed in their range during the workout of the intervention. And in the mild group, 2 subjects were above the target heart rate range, 2 out of 20. Here's the overall average heart rate by group during the intervention. And I think we largely achieved what we wanted to. Resting heart rate obviously is lower, and the differences between rest and mild and hard is about 40 beats a minute, which is what we were hoping for. So we think the intervention did what we wanted it to do. So in terms of the results for some of the cognitive tests, and I'm only showing you here the results for these first several tests for the 4 time periods at day 1. So it's baseline, 0 hour, 1 hour, and 2 hours. And the reason why I'm not showing the 24 hours for many of these tests is because we originally actually were not going to do the 24-hour reading on several of those tests. And during the 24-hour test, we only had 40 subjects versus 60 because we ran out of money. we couldn't do the last day on about 20 people. But for the Stroop task, though it looks like there's a difference in the hard group going down, none of these changes over time or the time by group interaction was significant. And here are the exact numbers. And the number that we analyzed was accuracy. So accuracy is what we call the number of hits, the time they got it right, versus the overall number of trials that they did. Here's the flanker test, which is the arrow test. There's a trend, a significant trend for the overall group to go down. And despite the fact that it looks like the hard intensity group responded differently, this interaction fell short, slightly short of significance. Verbal fluency task hits. How many words did they get when we asked them to do as many words in a minute? You can see it's essentially no change with exercise and no changes in any of the groups. This is for the visual short-term memory task, where those little colors appeared on the screen. Short time they do identify which changed. There was a significant trend down overall in the overall group. As you can see here from the numbers, the baselines were 46, 46, 43%. You can see it's a little bit, there's some lower values, but when you average it, it's down slightly. And again, it appears that the hard group did worse, but that interaction was just short of significance. For the food capture test, we're trying to distract people. There was a highly significant change for time, where the responses went down over time, but despite the fact that this looks much lower, it also fell short of significance. And this is the result for a long-term memory task, where subjects took the tested baseline, and then a day later, and you can see in all groups, it went down at a high level of significance. So what are our preliminary conclusions? So in 4 of the 6 tasks, there was a decrease in response over time. And though there were some trends for impaired cognitive function after hard intensity exercise, none of those were significant. We are waiting the results for the exercise effects on the blood markers, so I can't really tell you about it. What are our next steps in terms of analysis? Well, we're going to examine the habitual physical activity scores, the aerobic and strength fitness scores, and body composition in relation to exercise intensity and cognitive function. So we'll use those factors as adjusters in multiple regression analyses to see if, for example, maybe it's those people that were most fit at the beginning did better or maybe worse than those who were less fit. So that's what we're going to explore. And within the responses of the cognitive test, there are several other parameters that we are planning to look at. For example, reaction time. So individuals might have gotten the right answer, but did it take them longer after one of these conditions? And reaction time is important, because if that make a quick decision and you have a very slow reaction time, that's not so good compared to a quick reaction time. So we're going to see if the various levels of exercise interferes or helps reaction time. And then we're going to look at where we can, the results in terms of where the challenge is congruent versus incongruent, to see if there's a difference in that response. Shortly, we will be analyzing the blood markers, which are associated with brain function, and then and the long-term goal is really to expand this research to older subjects with and without signs and symptoms of cognitive impairment. As far as we knew, a study like this with the kinds of measures we made has not really done before. So that's why we started off with young, healthy subjects, one, to look at the feasibility of doing this, get some preliminary data, and the long-term plan is to look at other populations who are far higher risk for a cognitive decline. And with that, I'll stop, and I have a yellow light, which means I can probably take 1 or 2 questions.
Questions for?
All right. Okay. Oh, sure.
What were their fasting feeding states?
They could have a light snack, but no heavy meal at their discretion. Yeah. Even if they were hungry, we don't think that, it certainly wouldn't affect the exercise responses, but we did allow them to have a light snack. Sure. So as soon as we stopped the treadmill and just got the subject seated at the table where we had the computer set up, probably a minute or two. And these were healthy subjects, so we weren't worried so much about the typical warm-up that's recommended. So it was 0 hour, 1 hour later, and then 2 hours later, and then 24 hours later. And the testing itself probably took up most of that hour, maybe a good 30, 40 minutes. But yes. Yeah, that's a good point. You know, most of the tests. Oh, I'm sorry. So, is there a possible ceiling effect in some of these measures? Probably. We deliberately chose younger, healthy subjects who are most likely to do well on these tests, but we wanted to see, though, if exercise interfered. My guess is that if we do it in older people, we'll see a different pattern, at least in terms of their scores, and there may not be a ceiling effect. But we'll also soon have the blood to see how that changes, and because, you know, when you report results like this, you're reporting means, but within that, there's a lot of variation. So even though the mean is 50, you know, probably several people at 75 on some measure and a lot at 25, and you want to see who does what. And we're hoping to look at whether or not the blood and these other parameters of fitness and fatness also affect the response. Anyone else? All the way in the back. Man, can you just stand up so maybe and scream? Yeah. Although I didn't show the error bars, but those differences that you saw, at least in the hard group, that looked like they were a lot lower at some point, they were not far from being statistically significant. But you're right, there's some variability. I did not show the error bars.
Thanks very much.
Okay, thank you.
Kick us off.